Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. I hope all of you are well and fine. Today in this video we are going to see throat swab for culture and sensitivity test. If you guys remember, we already talked about different culture tests, like we talked urine culture, pus culture, culture, sputum culture, pleural culture, okay, acetic fluid for CS. So we already talked about these videos lecture related to the culture test. If you wanted to watch all these video lecture, you can go back to my channel and watch the culture test playlist, so you will find all the video lecture as I told. Okay, now today in this video we are going to see the throat swab for CS. So let's start the video. My name is Muhammad Kamil Khan and you guys watching my channel Kami Microbiologist. The main content of this video is that we are going to explain the introduction, then we will explain diagnostic purpose, culture media used for throat swab for CS, then we will see requirement and procedure, most common bacterial throat, you will see antibiotic profiling, most common biochemical test, and then we will see the precaution and limitations. First of all, let's introduce this topic. What is throat? Throat, also called as pharynx. If I show you the diagram, then I will explain it later. Okay, now this is the throat, we can say anatomy. This is the nasal area or nose area, which we can say nasal cavity. Now the air will come from the environment to the nose, and then it will be transfer and travel to the trachea, and then it will reach to the bronchus, bronchiolus, and finally to the alveoli. Okay, this is how the air can be moved. Now behind this trachea there is the esophagus, as you can see, which we call that the pipe for the food. This is the tongue area, this is the larynx area, or on the larynx area there will be on the larynx and esophagus area there is a cape like structure which we call that epiglottis. Okay, and there is also a structure which we call it tonsil as well. So this is all about the throat. Now if you wanted to collect the throat swab, so we have to take a swab, as you can see, and then we will touch the tonsil area. We will collect the sample from the tonsil, from the all surrounding of this throat area as well. Okay, this is how the throat swab. Now let's explain the topic. First of all, we have the throat or pharynx. Now throat or pharynx is what? It is actually the muscular tube that can connect the mouth nasal passage to the esophagus and larynx, as we talked. Then this throat can play an important role in both digestive and respiratory. Okay, now how? Because it is important in breathing, swallowing, and speaking. This throat can be divided into several parts, like we have nasopharynx, which is the upper part, oropharynx, which is the middle part, and laryngopharynx, which is the lower part. Moreover, this throat is also the home for the structure which we call that tonsils and epiglottis as well. These two things can protect the airway during swallowing. Okay, next we have the throat swab for CS. So it is the culture or medical microbiology test in which we collect the sample from the throat area. Here I write air, but it is by mistake. So we collect the sample from the throat area to isolate any type of the bacterial infection, and to find the appropriate antibiotic for their treatment. Diagnostic purpose of this test is to detect the pathogen. Culture for antibiotic sensitivity reveals which antibiotic agents are effective against the identified pathogen. Monitor the treatment response. The isolated bacteria during throat swab for CS is Streptococcus pyogenes 15 to 30% in children mostly, Streptococcus pneumonia 10 to 20%, Haemophilus influenza 10 to 20%, Moraxella catarrhalis 5 to 15%, Neisseria gonorrhea 1 to 5%, Staph aureus 5 to 10%, Klebsiella pneumonia 2 to 5%, and E. coli 1 to 3%. So these are the isolated organism and their percentage. Culture media. In the for the throat swab for CS, we use blood agar, McConkey agar, chocolate agar, MSA mannitol salt agar. Now for biochemical tests, if we wanted to do biochemical tests for throat swab for CS, let's suppose once the organism is isolated, so then we have to perform gram staining. From the gram staining, we get the idea whether the bacteria is gram positive or negative. Let's suppose if the bacteria is gram negative, we perform Vogas precursor tests and Dole tests. For gram positive, we perform coagulase, catalase, DNase test, oxidase, and API testing is also from the gram negative. So these are the biochemical test. Requirement is we need ethanol, wire loop, personal protective equipments. We need a burner or spirit lamp. We need culture plates. We need incubator, LFH, autoclave, antibiotic disc, and culture swab. Procedure is very simple. First of all, we have to collect the sample at morning time using disposable swab. Then we take a wire loop, will red hot it and perform the streaking. But before that, we have to make a base on this selective media as we talked, and then we perform the zigzag or quadrant streak method. After that, we incubate the plate for 24 to 48 hours for microbial growth. Then we will identify the bacteria using biochemical test. Once the bacteria is identified, then according to that bacteria, antibiotic disc will be placed onto the bacterial lawn. Then this antibiotic disc plate will be incubated for 24 to 48 hours to check the sensitivity, resistance, and intermediate of the microbes. Finally, then once the antibiotic profiling results obtained, then we will report the test. Antibiotic profiling. Now when you get E. coli, so you must use these antibiotic as I write. For Klebsiella, we use these antibiotic. For Staph aureus, we use these types of the antibiotics. For Streptococcus pneumonia, we use these types of the antibiotics. For Haemophilus influenza, these are the antibiotic for Haemophilus influenza. For Neisseria gonorrhea or Neisseria meningitis, we will use these type of antibiotics. For Moraxella catarrhalis, we use these type of the antibiotics, and for Streptococcus pyogenes, we will use these types of the antibiotics. Okay. Precaution is very simple. You need to collect or you must collect the sample during morning time. You have to use sterilized disposable swab. You have to clean the working area before and after use. You have to process the sample within 1 to 2 hours immediately, otherwise you store the sample into refrigerator. You have to label the sample. You must use quality control and appropriate culture media. You must do gram staining and biochemical test to confirm the bacterial growth. You have to use appropriate antibiotics and follow the CLSI guideline for the measurement of the zone. You have to guide the patient to stop the antibiotic before 1 week. You have to avoid the contamination of bacterial and fungi, and you must ask patient history and age, sex, etc. Limitation is that in this test the patient feel uncomfortable. In this test the contamination may also be occur due to the skin normal flora. In this test once the patient using antibiotics, so due to this way the culture test will be negative generated, and it can generate a negative result due to the bacterial growth suppression by using antibiotics. It takes 72 hours, 24 hours, which is the long time. It is expensive method. It required a high special techniques. It is a big challenges for everyone to differentiate between pathogenic and non-pathogenic organisms. In this test different techniques can be ignored for the fastidious organisms, so due to this way negative report will be generated. So this is all about the throat swab for CS. Thank you so much for watching. If you like this video, make sure to hit the like button, share this video, and subscribe this channel to get more interesting videos like that. Thank you so much for watching.